Hello everybody, this is part 2 of subtopic 6.3, Defense Against Infectious Disease. Last time we left off talking about non-specific immunity with these phagocytic leukocytes. So leukocytes is a general term for white blood cells, sort of like erythrocytes is the term for red blood cells. So if something is phagocytic, then it would be something that goes through phagocytosis, which is a type of endocytosis, where one cell is engulfing something else and engulfing it and bringing it into itself and then digesting it using the enzymes. Here's a really cool video of a phagocytic leukocyte chasing a bacterium. And then you can see it finally does engulf the bacterium and bring it into itself. And then it starts to digest it using a lysosome, and the digestive enzymes break it apart. So another term for phagocytic leukocytes is just phagocytes. And they do move in response to some chemicals, and that's how they're actually chasing the pathogen. So some of the things that they use to chase the pathogens are proteins produced by the pathogen and phospholipids released by the damaged cells. This could also be a good opportunity to review some of these concepts about the plasma membrane and vesicles, membrane fluidity and fusion, and endo and exocytosis. For example, with membrane fluidity and fusion, you could be thinking about the vesicles, which are the lysosomes. Lysosomes are vesicles of digestive enzymes, right, that break down the pathogen. So those vesicles actually fuse with the membrane of the phagocytic leukocyte, and then they take the pathogen inside of itself through endocytosis. And then don't forget to refer back to the statement whenever you're studying as well, because that's the core of what's really important. So ingestion of pathogens by phagocytic white blood cells gives nonspecific immunity to diseases. So this is part of the nonspecific immune response of our bodies. As a quick review, our first line of defense against pathogens is our skin and mucous membranes. This is also technically non-specific immunity. Why is it non-specific? Because it's not targeting any specific pathogen. It's a way to protect from all pathogens. Another line of defense against pathogens is our ability to use clotting factors that are released from platelets to seal the skin with blood clotting. Blood clotting allows us to keep that skin barrier from the previous slide continuous so that there's no openings for pathogens to enter. Quick review of blood clotting is that it's a series of chemical reactions that occur to seal up a wound. So first there's platelet or cell damage. Clotting factors are released from the platelets. Prothrombin gets converted into thrombin due to these clotting factors that are released. Thrombin turns fibrinogen into fibrin, and fibrin is that sort of mesh that forms over the open wound, and it also captures the erythrocytes, adding to sort of the clumping and clotting of blood. If the pathogen happens to pass the skin and mucous membrane barriers of nonspecific immunity, the next phase of nonspecific immunity is these phagocytic leukocytes, the phagocytes, which would eat and engulf the pathogen and then digest it. Again, this is nonspecific, so a phagocyte is going to attack anything that it recognizes as a pathogen or a non-self cell. Okay, so now that we spoke about nonspecific immunity and what that's all about with the phagocytes, we're now going to shift over to another type of leukocyte or white blood cell called lymphocytes. Lymphocytes are involved with specific immunity and producing antibodies. So some more specific names for these lymphocytes that are involved with specific immunity are B cells, T cells, and killer cells. For this subtopic, 6.3, you only really need to know about phagocytes and lymphocytes. However, if you are an HL, then you are going to need to know more details about B cells and T cells. The next statement reads, production of antibodies by lymphocytes in response to particular pathogens gives specific immunity. So this statement is describing how those special leukocytes called lymphocytes produce antibodies in response to particular pathogens that give specific immunity. So what does this mean? Antigens are markers on the outside of cells. All cells have these antigens. Even your red blood cells have these antigens, unless you're blood cell type O. Okay, so a pathogen has antigens on the outside of the cell, on the cell wall or cell membrane. Antibodies are produced by the lymphocytes in response to detecting the antigens on the outside of the pathogen. Now, how is this different from non-specific immunity? This is specific immunity, meaning there are specific antibodies that match with specific antigens. Notice that not all of these antigens pictured here would fit correctly into this particular antibody. Specific immune responses are triggered by non-self cells. What's the difference between a self cell and a non-self cell? Well, a non-self cell would be considered as a pathogen. Is it always going to be an actual pathogen? No, but it's a cell that your body would recognize as not being of yourself or your own body. And how does it recognize that? With the antigens on the outside of the cell. Versus self cells, which also have markers or antigens on them, but the lymphocyte pictured here is smiling because it knows that this is a self cell, so he's not going to attack the self cell. Here's one that is angry and frowning because it is going to attack that non-self cell because it's recognized it as a pathogen. So we've talked about antigens, their corresponding antibodies. We've talked about self cells and non-self cells, but we haven't talked about the production of antibodies. How does that all work and how does it happen? So first we start with an antigen that would be on the outside of the pathogen, antigen X. These are all lymphocytes pictured here, A, Z, B, A, Y, Z, C, all different lymphocytes. But only lymphocyte X has the receptor on the outside of the cell that detects antigen X. Once it detects antigen X and knows that it can produce the antibodies that correspond to antigen X, it's going to clone itself and make multiple copies, thousands of copies of itself, and this is part of the specific immune response. It's then going to produce those antibodies. What do the antibodies do? As we said before, they match specifically to the antigen on the pathogen and then mark it for destruction. Sometimes they render the pathogen useless, so the pathogen can no longer function, and then they also do mark it for destruction by a phagocytic leukocyte. Here's a simple yet effective animation that shows a pathogen in the center here, possibly a bacterium, with its antigens on its cell wall. And then you have a lymphocyte over here on the right, you have a lymphocyte over here on the left. Notice that these lymphocytes are not the same. Do you notice what's different about them? If you guessed that it was the antibodies, the antibody receptors on the outside of the cell, then you would be correct. So let's have a look at the animation and how this all plays out. As you can see, the lymphocyte on the right had the correctly matching receptor on the outside that matched with the antigen on the pathogen. It then starts producing the antibodies, thousands of antibodies, that are sent out and attached to the pathogen and mark it for destruction. As a recap, this is the process of specific immunity. Is it specific? Because specific pathogens are being targeted by specific lymphocytes, and specific antibodies are being produced by the lymphocytes to attach to the specific antigen and destroy that specific pathogen. Maybe you've heard the phrase that a person can become immune to a pathogen. This is because this is called immunity. This is specific immunity. Some of those cloned cells, the cloned lymphocytes that then produce the antibodies to render the pathogen useless and mark it for destruction, some of those cloned cells will remain long after the pathogen has invaded the body. And why does it remain? Well, just in case that pathogen re-enters the body and the body has to use its specific immune response again, but this time it can attack the pathogen much faster than the first time that the pathogen entered the body. Vaccines actually work under the premise of specific immunity. The way that vaccines work is they jumpstart your body into creating immunity for a pathogen. Vaccines contain weakened or damaged forms of the pathogens, and then those pathogens contain antigens which your lymphocytes would detect and then start producing the antibodies. The next statement reads, antibiotics block processes that occur in prokaryotic cells but not in eukaryotic cells, and viruses lack a metabolism and cannot therefore be treated with antibiotics. So the key idea here that the IB wants you to realize is that antibiotics are drugs used in the treatment and prevention of prokaryotic bacteria. So they are not used to treat viruses, and they are not used to treat eukaryotes. You need to be able to explain why they cannot be used to treat viruses and eukaryotes. The answer would be that antibiotics are designed to disrupt structures or metabolic pathways in bacteria and fungi only. How are they only disrupting structures or metabolic pathways in bacteria and fungi and not in viruses and not in eukaryotes? Well, their cell walls and their membranes are significantly different than the cell walls and membranes in eukaryotes. And their protein synthesis is very different as well. Their DNA and RNA synthesis is different because they have the 70S ribosomes versus eukaryotes, which have the 80S ribosomes, and other metabolic processes like differences in enzyme function. None of these things even exist or are very different in viruses, and in eukaryotic cells, some of these things exist, but they're very different. So antibiotics have no effect upon them. So again, viruses lack a metabolism. They don't do any chemical reactions within their own components. They hijack other cells' metabolism and use those for their benefit. This is one of the reasons why viruses are not even considered to be living creatures, because they lack a metabolism and cannot therefore be treated with antibiotics. Some strains of bacteria have evolved with genes that confer resistance to antibiotics, and some strains of bacteria have multiple resistance, and we're going to talk about that in a second. But first, we're going to talk about why would you take antibiotics for cold and flu? There would be no point, because colds and flus are created from viruses. Okay, so if your doctor prescribes you antibiotics and you have a cold or flu, there's no reason to be taking it. In fact, sometimes it's overprescribed, which is part of this statement and why they're saying some strains of bacteria have evolved genes that confer resistance, because the reason why they've conferred resistance and why antibiotic resistant bacteria have developed is because of the overprescription of antibiotics by doctors. That's what this slide is all about. It's talking about how there's been indiscriminate use of antibiotics because it's been overprescribed, and it's leading to antibiotic resistance in bacteria. In some countries, you can even get antibiotics very easily over the counter. This is a problem because it's creating antibiotic-resistant strains of bacteria in which that particular many antibiotics may now not be effective against bacteria. So this is all related to natural selection and evolution because bacteria are exposed to the antibiotic. If people don't finish their whole round of antibiotics, which also can happen, or even if they do finish their whole round of antibiotics, sometimes the bacteria within the body does mutate and become resistant to that particular antibiotic. That bacteria that then mutates and becomes resistant will divide rapidly and create a new strain of bacteria that is now resistant to whatever antibiotic was taken by that person. Okay, so here's a review slide from 5.2, Evolution of Antibiotic Resistance in Bacteria. And here we've got the original bacterium. They each have their own variation that was produced. These ones right here are susceptible to being killed by the antibiotic. As you can see, the antibiotic or antibody attached to the antigen on the outside of the bacteria. However, this one had different antigens or some kind of mutation that made it resistant to that antibiotic. So then it keeps replicating and develops that new strain. And then here we have a very specific example with MRSA, which is methicillin resistant. Okay, so the original strain was methicillin susceptible, and methicillin is the actual antibiotic that gets applied, and that the methicillin susceptible bacteria is susceptible to being killed by that, but then a mutation occurred in that particular bacteria, and then it became methicillin resistant, or MRSA. So the concern with developing antibiotic-resistant strains of bacteria is that there could be sort of a post-antibiotic era where we've run out of all possible antibiotics that can be used, and then there's a revival in certain strains of bacteria that are all resistant to antibiotics. And so this is a major concern of doctors because strains of antibiotic-resistant bacteria are increasing more and more. One of the reasons why scientists and doctors are so concerned about antibiotic-resistant bacteria is, as I was saying before, the number and diversity of new antibiotics is actually decreasing. The number of effective new and approved antibiotics is decreasing drastically due to this antibiotic-resistant bacteria. Okay, so now we have a specific example of the first antibiotic that was created by Florey and Chain's experiments, although another scientist is behind all of this as well. His name was Fleming. Here's a kind of a cool video of some original footage of him when he first realized that penicillin is an antibiotic.
In one of the glass dishes where he cultured germs for his experiments, Fleming noticed one day in 1928 that some mold, such as appears on decaying food, had begun to grow. A spoiled experiment. But with mind alerted by the earlier work on protective substances, he looked closer. He saw that near to the mold, no germs were growing. Might it be that this mold, like the human body, produced a substance capable of destroying germs? He made tests and found that even some of the most dangerous germs died near to the mold.
Okay, so he discovered the effectiveness of penicillin as an antibiotic, but he was not able to purify it enough so that it could be used on animals. But then these other scientists, Flory and Chain, um, assembled a team of scientists to try to actually purify penicillin enough so that it could be used on mice. And then they were able to use it effectively on mice, and they then figured out a different way to purify it so that it would be effective and non-harmful in humans. So this is also a nature of science statement, so sort of like a TOK tie-in, that Flory and Chain's tests on the safety of penicillin would not be compliant with current protocols on testing. So this is an outline of our current modern practices on drug testing and our current protocols. They did not always follow those protocols when they were testing the effectiveness and the safety of penicillin. Now we're going to shift back into viruses, specifically HIV. So this statement reads, the effects of HIV on the immune system and methods of transmission. The effects of HIV on the immune system are significant and devastating because it's a retrovirus. Okay, so a retrovirus, what they do is they insert their own viral DNA into the host cell and then incorporate that DNA into the body's host cell's DNA. And specifically, it attacks the helper T cells, which is a very big deal because that's how it attacks the immune system. The helper T cells are involved in antibody production. So essentially, HIV is attacking the lymphocytes that produce antibodies, and that's why it's such a deadly disease. So one of the effects of HIV, human immunodeficiency virus, is that it leads to AIDS, which is the acquired immune deficiency syndrome. Okay, and so it does take sometimes years for AIDS to actually develop. The way that this all works is there's a primary infection in the beginning where, as you can see in the beginning, there's a healthy level of these T lymphocytes. So these are those helper T cells we were talking about, the lymphocytes that help to produce antibodies. There's a healthy number at the beginning of the infection. And so then as we move on, as time moves on, the HIV keeps copying itself, and copying itself. And the T lymphocytes do fight it off at first and is able to kill off some of the virus, but then eventually, because that HIV, the virus, is attacking those lymphocytes and continuously replicating within the lymphocytes and killing them off, there reaches a point where so many of those lymphocytes, which are part of specific immunity of all kinds of pathogens, right? When they start dropping off, those levels and numbers go down to a critical level. That's where acquired immune deficiency syndrome develops, and the body no longer has a healthy immune system because too many of these lymphocytes have been killed by the virus. And then the virus continues to spread and spread and spread until all of the T lymphocytes have been depleted and the person no longer has an immune system. This is part of why, if you've ever heard people say that HIV in and of itself is not the thing that ends up killing a person with HIV, but rather it's their lack of and immune system. So their lack of ability of specific immune response where they're able to produce antibodies. Okay, last part and last statement is the methods of transmission. So what are the risk factors of HIV transmission from no risk to lowest to highest? This is what they are. So essentially, skin contact, toilet seats, mosquitoes, no risk at all, because there would be no transmission of cells that contain HIV.